So today, I've been asked to provide at the patient level, at the family level, a deeper understanding of the disorders. And really what I wanted to do was to provide a straightforward way of describing them. You've all heard this information many times yesterday. So if you're comfortable with these pathways and disorders, I'm still out of breath from running, I'm sorry. Then, just chill, look at your phones, do that sort of thing. But if you want to have a bit of a revision, this is what this is going to be. So here is what I want to talk about today. I've been asked to describe what is creatine, why do we need it? How do we make creatine? Why are there 3 disorders? How do we treat them? And why are the treatments different? So just to start off, you've already seen this picture of Dr. Salomons showed yesterday, the French chemist, Michel Eugene Chevrel. So we've known about creatine for nearly 200 years. It's a small molecule, it contains nitrogen, and it's mostly found in our muscles. 90% of it is in our skeletal muscles. So how do we use it in the body? We need it to maintain our chemical energy in the cells. Chemical energy is adenosine triphosphate, otherwise known as ATP. Creatine is converted to creatine phosphate, and creatine phosphate is used to make ATP. So these are the structures of creatine and creatine phosphate, and you see that the difference is this red circle here shows the phosphate group, and together creatine and creatine phosphate make the creatine pool within our cells. Creatine and creatine phosphate spontaneously cyclize to creatinine, which is not useful to the body, so the body excretes it in the urine. And we lose a small amount of our creatine from our cells every day, which is why we need to replace it. So energy production in the cell starts with the mitochondria. So this is a little example of a cell, and you see here is the nucleus where the genes are, and then we have what are known as mitochondria. And the mitochondria are like the battery packs within our cells. So this is just a closer view of the mitochondria. So inside the mitochondria, we start with the molecule adenosine diphosphate. Di means 2. There are 2 phosphate groups attached to adenosine. What the mitochondria do is take the fuel from our food, the glucose, the fats, the proteins. They convert that energy captured in that food and use that to add a phosphate to adenosine diphosphate to make adenosine triphosphate. And then, still within the mitochondria, the ATP, which is now an active form of chemical energy, is used to transfer a phosphate group to creatine, to make creatine phosphate. So here, this is where it all starts in the mitochondria, and then creatine phosphate is transferred to other parts of the cell where it can be used. So from the mitochondria, creatine phosphate is transported, and then in the cytosol, which is the soupy part of the cell, you have adenosine diphosphate again. So adenosine diphosphate is not useful for energy. It's not an energy source, so we need to make an adenosine triphosphate in the cytosol where it is going to be used. And the enzyme that does this is creatine kinase. Enzymes are catalysts. They drive reactions. Without enzymes in our cells, these reactions will not happen. And then adenosine triphosphate is used in many different ways by the cell. And the energy that is derived from cleaving that phosphate, the third phosphate from ATP, is used to drive the reactions. So that is a very simple overview of why we need creatine. So as you have heard yesterday, Creatine is made in different parts of the body. And to be honest with you, it sounds like we don't fully understand the whole story of creatine synthesis. We know that the liver, kidneys, and pancreas are responsible for a large part of creatine synthesis, but we also know that it probably happens in the brain and other tissues too. So creatine, if you're an omnivore, if you eat meat and fish, you'll get about 50% from your diet. But if you're a vegetarian like me, you have to rely on your own body to make it. So vegetarians tend to have lower creatine. And then why do we need to either make it, our bodies make it, or replace it by the diet? It's because we're losing creatine every day from our cells. So the synthesis pathway that our bodies perform starts with two amino acids, arginine and glycine. Now, amino acids are the building blocks of proteins, and some of them our bodies can make, and some of them we need to get from the diet. So glycine, our bodies can actually make glycine, so it's not considered essential in the diet. Arginine, on the other hand, is considered semi-essential. Adults have no problem making it, but very young infants may need an exogenous source of arginine, get it from the diet, because their pathways for making arginine are not yet mature. So the first enzyme, AGAT, what does it do? It takes a little piece of arginine and it sticks onto glycine. It takes a piece called the amidino group and it transfers that and adds it to glycine. And as a result of that, ornithine is made. And guanidino acetate, of course. And then guanidino acetate is converted by GAMT to form creatine by taking a methyl group. What is a methyl group? It's just a one-carbon compound. 1 carbon, 3 hydrogens. Our bodies use methyl groups in many different ways. We use it to switch on and off genes. We use it in reactions such as this. And the source for that methyl group is a compound called S-adenosyl methionine, SAM for short. So our bodies make SAM. SAM provides that methyl group, and then our bodies use that to make creatine. And quite a lot of the SAM that our bodies make is actually consumed by this pathway. A lot, at least 40% or more. So once you've made creatine, then you've got to get it into the cell. Now, as we have heard yesterday, there's a concentration gradient. It's low concentration in the blood, high concentration in the cell. And now this is a problem because the laws of physics and chemistry are such that chemicals like to move from high gradient to low, from high concentration to low concentration. So trying to get it up that hill requires a form of energy. That energy that the cell uses is a sodium gradient. So if you think of the funiculars, which are little trains that can take you up a steep slope, you always have a counter train coming down as one train is going up. So the counter train coming down is that sodium gradient, and that helps drive the creatine get into the cell. So creatine is taken up from the blood. It's also taken up from the filtered urine in the kidney. So when your blood gets filtered by the kidney, and the fluid enters the tubule in the kidney, that's the first part of urine production. And there are a lot of transporters in the kidney to return back to the blood things that are useful to the body. Creatine is one of them. So this is what the creatine transporter is doing, is returning back to the body the creatine from the urine that's filtered out in the initial stage of urine production. There's also a high probability that that creatine transporter is needed to take creatine back up from the CSF back into the neurons, and this was discussed yesterday in some of the talks. Now, where does the creatine pathway exist? We know that there are some tissues where it's well characterized. It occurs in the liver, kidney, and pancreas. But there's also been research to show that some of it can happen in the brain, in which cell type. There's a little bit of confusion. So looking at there's a website called Protein Atlas, and what this is, it is a database that lists what we actually know currently about protein expression, and it's measured at the level of the protein. So they have an antibody against the protein, and they look at different tissues, and they measure the quantity of protein in those tissues. They can also look at the messenger RNA, which is an in-between molecule between the gene and the protein that the cell produces, and this is a measure of expression of that gene. So looking at both those pieces of evidence, they have shown that there's high expression of the first enzyme, AGAT, in the liver, kidney, and pancreas, and a medium amount of expression in the brain and in the intestinal tract, in the stomach, the duodenum, which is the upper intestinal tract, and the colon. Whereas the GAMT protein has medium expression in liver, kidney, pancreas, gallbladder, and the GI tract. So we know that, let me just see what I'm trying to say here. Lost my train of thought, sorry. Creatine expression, the creatine pathway occurs to some extent in the brain, but just how, obviously that is not enough to protect the brain against a deficiency when you can't transport creatine into the brain. So the creatine transporter still plays an important role to maintain those levels. So the disorders are well defined. We know you start off at the gene level with changes in the gene. This causes a problem with making the proteins, and then you end up with the 3 different disorders. The main problem of this, of course, results in deficiency of creatine in all tissues, but the problem in the brain trumps the rest of the other problems. The problem in the brain and the most severe consequences clinically for these patients. What do we know about these disorders? Well, we know what has been published, and it's families like yourselves which have generously allowed sharing of this clinical information in case reports that allow us to better understand. And this is why there is such a, why there a call to arms for families to help with the research, and that starts with sharing information about the diseases and how it affects health. So we know it's well described already in the previous talks yesterday that the neurologic impacts are the most severe in developmental delay in young children, and then quantified as intellectual disability through IQ tests in older children and in adults. And then you have the global speech delay that occurs in virtually all patients, and then more variable presentations of the behavior, the abnormal movements, the seizures. These happen in a lot of patients, but not in all of them. And then the striking low muscle mass that was shown by Andreas yesterday, Dr. Schultz, in his patients. And the muscle story does tend to be a little bit overlooked, I think, because we're all focused on the neurologic. Some carrier females for the transporter may have no symptoms at all, and then they may have mild learning problems, or they may present as with a severe phenotype like the males. And then, in the creatine transporter, there is deficiency. There is reports of gastrointestinal problems, vomiting, diarrhea, and constipation. And actually, they share this feature with mitochondrial disorders, another problem with making energy. So the GI tract is very vulnerable to low energy sources. So the treatment goal is trying to explain now why there are 3 different treatment approaches for the 3 different disorders. Obviously, the primary goal is to get creatine into the brain. And it's by just providing dietary creatine, we can bypass the blocks in the first two disorders. And the case reports have shown the benefits of doing this, especially pre-symptomatically, before there is cognitive delay and speech delay. This is very effective. But if you do it after these have already manifested, you're unlikely to reverse some of the damage. So this is why newborn screening for these disorders is so important. And the other thing I want to like to mention is that a lot of the studies that are published are retrospective, and they're not systematic. So we need better prospective, systematic studies to study the natural history. So the creatine transporter, you don't bypass the block by giving creatine, and this is why it is relatively ineffective compared with the first two disorders. If you have an attenuated form, which means you have some residual activity, and most patients do not, but as Dr. Longo reported yesterday, there was one patient who had 30% activity in fibroblasts, then in those patients, there is a chance that giving creatine will make some improvement. And then in females who not every cell is impacted because of random X inactivation, some cells will express the good transporter and some will express the impaired transporter. And so the cells that we're relying more on the cells that are expressing the good transporter to compensate for those cells that are not working in females. So in females, there is more chance that they will respond to therapy. And then again, so this is why there is such big interest, and I'm very excited to see so many different companies now working on new approaches to treatment for the creatine transporter. Supplementing arginine and glycine in the diets has been tried, and subjectively and retrospectively has been reported to help some patients. And this is trying to maximize these pathways, the activity of these pathways, especially in the brain. So in guanidinoacetate methyltransferase deficiency, we of course have the problem of guanidinoacetate accumulation, which is neurotoxic, so we must reduce it. Dietary creatine on its own has some negative feedback on the AGAT enzyme, and this helps reduce guanidinoacetate, but it can't do it. It doesn't reduce it all the way, so we need to do more. Reducing arginine in the diet by low protein diets, and then to remove glycine with sodium benzoate. And we had a lot of discussion about the pros and cons and the relative efficacy of doing all of these things. But I think the most striking impact on the AGAT activity appears to be ornithine. Ornithine has negative feedback like creatine and seems to really help get that guanidinoacetate down to a much more mild level in the blood. So just to summarize, we need creatine to maintain our chemical energy in the cells. When you can't make creatine or take it into the cells, this causes a problem for many different parts of the body, but the brain manifestations are the most serious. The transporter is challenging to treat, and it's unfortunately the most common of the three because it's X-linked. And in AGAT and GAMT deficiency, the treatments are very successful if started early. So that was the first part of my talk, and I've also been asked to talk a little bit about ClinGen. We wanted actually a representative from the ClinGen program to talk through WebEx or to come here, but unfortunately we got hooked up with them a little bit too late to make that arrangement for this meeting. So what is ClinGen? So ClinGen is funded by the NIH, and it is a research program to understand how gene variants affect health, and this is across many different disorders, not just the creatine deficiencies. So how can you share your data? So the ClinGen researchers are asking different parties to participate, be they laboratories that perform testing, that identify the variants, be they the patients and the patient groups, such as the ACD, and then also clinicians. And then to share this into a publicly available database. This the intention of the ClinGen researchers to then take this data and learn more about how genetic changes impact health. Why would we want to do this? Well, quite often, I mentioned in my talk on diagnostics yesterday, when a variant is identified in a gene, quite a lot of the time, we don't know what that variant might do. We can make predictions, but it may never have been found before and reported. And so to know what it does, it can be challenging, and then to provide an accurate interpretation. So there are some changes that we have that are well described in genes, but we might not know everything about them. So by sharing more data, we can learn even more. We want to increase the understanding of disorders by sharing our data, and it will also help people who are being newly diagnosed, if they are diagnosed with the same gene variant that your family has. By sharing your data, you can help them. And then in the future, it's possible that research will be done on the data set, and then that data can be then used to help your family understand your condition better. So it's a circular process, and it helps in many different ways. So ClinGen have an appeal to patient groups. We can't do it without the patients. We can't do it without the willingness of patients to share your what we call private health information. We as laboratorians and clinicians can't just take your data without your permission and share it. You are the gatekeepers of your own private health information, and it's up to you whether or not you want to share. So how can you do this if you're interested? We have a patient registry for creatine deficiency disorders, and this is a partnership we have through Invitae. Invitae are a genetic testing company, and they provide a freely available, no-cost patient registry that they call Patient Insights Network. And ACD have been very, very rapidly got this together, and now there is this registry. And I'm very pleased to have seen all the hands up when asked who is in the registry. That's wonderful that you're already committed to this registry. So what ClinGen are doing, they're partnering with Invitae to through the registry, capture that data, and then put it into a public resource. There are other ways, so we have a registry, and this is how we hope that the patients with creatine deficiency syndromes will share their data. What about other disorders? Well, as individual patients, you can do it through a generic registry called Genome Connect. And then other patient groups can get organized and actually develop their own registry. But we're already ahead of the game. We were told by Jules Savat, who is one of the ClinGen researchers at Geisinger. These are, I should acknowledge, actually, these are her slides. That she has said that we're one of the first groups to be poised, ready to share data. So that makes us ahead of the game a little bit, and it's very exciting that we can do this. So what happens? There is that you have to opt in. We can't just take your data. You have to choose to share your data. And there will be more. I don't exactly know how this is going to be rolled out. We are still discussing that, trying to formulate that, but this will be coming your way in the next year. And once you choose to decide to share your data, what it means that data in the registry will be taken, and the ClinGen team will curate that data and put it into a database. Additionally, we want to capture information about health, so there could be questionnaires that are sent out. And these questionnaires will be sent to you, and fill it in, and then we will have that entered into the database. And again, this is entirely voluntary, and if you don't want to do this, you don't have to do it. So what is ClinVar? ClinVar is the database where the data will be put. It's publicly available. In fact, I used it for my talk yesterday. I was curious to know how many variants had been entered into ClinVar and how they'd been assigned as pathogenic, disease-causing, or non-pathogenic. I wanted to include some of that information in my talk. I was able to just go online and look it up. And so any of us can go and have a look and interrogate. And this is the great thing about it, that it is publicly available. So once the data is in there, we have a team of people who help put the data in there and make sure accurate information is put in there. So they are the curators. And then the next stage would be to form an expert panel. And an expert panel will then start looking at individual variants and scoring them, giving them a star rating. The higher the star rating, the more convincing that the evidence is that this variant is disease-causing or not. So this will really help the identification of new patients and avoid diagnostic confusion in that process. And it might also help with research, especially research that might be targeted to, say, missense mutations. So that's the end of my talk, and I'd be happy to take any questions.
I've got one real quick basic question, and at risk of making myself sound completely uneducated about it. So with the creatine deficiencies, the cell doesn't get creatine, to put it really simply.
Right.
So what does the cell run on? Because all the cells don't die. Is there, and I know this is really simplistic, but has there been research into figuring out what the cell does run on and supplementing with more of that, not as a cure, but as maybe a 5% solution, maybe bump it up 5%, help a little bit?
What does the cell run on when it doesn't have creatine? Andreas, can you answer that?
I don't think it can run without creatine. It's ATP. I mean, it's still ATP, it just is reduced.
Okay.
So it functions, it just doesn't function as well as a normal cell with creatine blood. So there's still ATP, there's still mitochondrial production. There's actually a little bit more mitochondrial output in these cells. It's still running off of ATP just like every cell in the body has. It just can't replenish it as quickly.
But I mean, it's not working with zero creatine. There's no model, there's nothing. So if you run a cell with no creatine, that's impossible. But with the ATP, it has very high energy. You cannot get ATP from the mitochondria to the cytosol without losing the energy. This is why you need creatine. It's only a transport system. We don't have any example where a cell works without creatine. There's only one alternative because this creatine system exists since we have the animal kingdom. This is it's 1,000 million years, and the only alternative to the creatine creatine kinase system is arginine kinase. So that's a similar system, but it's not existing in humans. So the spunk is, they can do this in some other living species, but not humans.
That's a great presentation. Thank
Just
you.
one more question on the ClinVar publicly available database. I just want to make sure, I think I know the answer to this, but just for everybody's clarity, the publicly available data is scrubbed, right, for names and...
Oh, yes, I'm so sorry. Yes, it's completely de-identified. Okay. Yeah.
I just thought that was important to point out.
Thank you, yes.
Any other questions? Really quick, Dr. Longo's getting his mic. Sarah, if you want to, just anybody else? We don't have an open panel today for discussions, so if you do have questions, now's the time. But I thought that was a wonderful, wonderful presentation.
As the next speaker is getting set up, so you mentioned for CTD, supplementation with creatine arginine has, it's not there yet, but maybe there are some improvements. Maybe not statistically significant, but reported benefits. So I guess my question is slightly different, though. It's not on whether there are benefits or not, but if kids with CTD are being supplemented with creatine and arginine. You mentioned that for a different case, there was this problem of GAA being created, right, which had its own problems. So if kids with CTD are supplemented with arginine and glycine, will there be a buildup of GAA for them too, and should something be done to actually counteract that? Is it creatine that's supplemented that will actually counteract that?
So creatine can be supplemented in creatine transporter to try and help get it into the cells somehow. Or you can try and boost the endogenous synthesis of creatine by giving arginine and glycine. And your question is, would that cause an elevation of guanidinoacetate, which is neurotoxic? And the answer is no, because in the creatine transporter patients, they have functional GAMT. So they have no problem converting GAMT to creatine within their cells. They can make creatine, they just can't get enough of it into their cells. Does that answer your question?
So even with the additional dosage, whatever GAMT is present in those kids will
Right. It's hard to get the guanidinoacetate to a level where it's a problem. And we can see this in the patients that are given arginine in urea cycle disorders. They might have mild elevations of guanidinoacetate, but not at the level where we think it's really causing a problem. Yeah.
So I have a question too. So in the list you had about the endogenous process for creatine, muscles weren't listed, right? So it seems like muscle cells do not produce their own creatine, right? And I think this comes back to Dr. Schultz's question yesterday of, how come muscles seem to be doing relatively well compared to?
Well, I just took a big overview of the expression because there were more than 20 tissues listed, and I didn't want to overwhelm. There might be a small amount of production in the muscle, but it be at a low level. Yeah. So.
And I guess we don't know how come muscles are doing so much better.
Well, are they? Because we know that the patients have some muscle weakness. We know that there is low muscle mass, but maybe clinically we're more focused on the brain issues. So, I mean, I don't know. An MD in this room can probably better answer that question.
Well, we don't really know about muscle transport. So we had 2 patients that had normal creatine amount in muscle, indicating that there must be another transport mechanism for muscle. I don't think anybody has shown a decreased amount of creatine in the transporter deficiency patients.
But in the creatine synthesis disorders, isn't it true that there is low creatine in the muscle, right? But that's because they're not getting it from the main sites of production. So, yeah.
I would like to go back to the question of ornithine and arginine supplementation in the creatine transporter deficiency, because I don't think that's a trivial question. I mean, you are perfectly right that transporter patients have GAMT, so they can, in theory, produce creatine from GAMT. But if this was really the case, I mean, this is, of course, what we say, but if that was really the case, then there would be no creatine deficiency. The problem is that although creatine transporter patients do have GAMT, they are not able to use it. The brain does not use enough GAMT. So, and still 59% of transporter patients do have seizures, and that may be due to accumulation of guanidinoacetic acid. And in fact, in Bressan's scheme that you all know very well, guanidinoacetic acid is made in glial cells, but then it needs to be transported into neurons to be changed into creatine. And neurons need transporter to uptake it, and they don't have it. So GAA does accumulate in creatine transporter deficiency. So I don't think it is a bad idea to supplement ornithine to these patients to try to keep lower the guanidinoacetic acid in this case. And I think it is a tricky question that we don't know perhaps so well, I think.
Could we do more research?
Yes.
Yeah.
Absolutely.
So,
And I would also give some ornithine to transporter patients, just in case.
Thank you, Dr. Young. Thank you.